Welcome to Unit 4, Lesson 2. Today we're going to be discussing the eukaryotic cell from the inside out. And by the end of this lesson, you should be able to compare and contrast the structures of plant and animal eukaryotic cells, and be able to recognize and describe the structures and functions of various organelles found within human cells. This lesson actually isn't intended for you to sit down and watch start through finish. It'll be quite long, so there'll be several breaks throughout, and you can decide how you approach it. To begin with, we need to recall what is a eukaryotic cell. So the question is, do you remember what "eu" refers to? Hopefully that's right. It means true. And what does karyotic or karyon refer to? That would be the kernel, which in this case is nucleus. So eukaryotic cell was originally thought to have a true nucleus. Unfortunately, we found some examples of eukaryotic cells that lack a nucleus, for example the red blood cell. So over time the definition has morphed, and today a eukaryotic cell typically refers to cells that contain what? You said membrane-bound organelles. You are correct. They have everything put away in nice, neat little places called organelles. Of course, that's a great definition, as long as we know what an organelle is. So an organelle is a distinct recognizable structure within a cell that performs a specific function. And a way to understand an organelle is simply an organelle is to a cell what an organ is to the human body. So it's a smaller part of the cell that has a distinct function, just like the heart is a smaller part of the human body that has a distinct structure and function. And just to quickly overview the organelles of plants and animals, we're going to compare and contrast what's found in an animal, what's found in a plant, and what's found in both. And I find it easiest to work from sort of the inside out. So in both we have a nucleus, which is attached to an endoplasmic reticulum. Which is abbreviated ER, which is attached to a Golgi apparatus, indirectly attached, which is then connected to vesicles, which are related to vacuoles, and in the similar size scale you have lysosomes, which actually recently have just been identified in plants, peroxisomes, and then slightly larger than that you have mitochondria, Down at the opposite end of the size scale, very small, you have ribosomes. And then you have the cytosol and the cytoplasm. I'm going to write cytoplasm first because we're running out of room. Oh, and we missed one thing that's also throughout, so we'll sneak it in here. Bad planning on my part. The cytoskeleton. So we're going to talk about all these organelles. You can understand how it's going to take a little bit of time, but it's pretty interesting. Over here on the plant side only, this thing that is green in color, hopefully you recognize as the chloroplast. Now I don't mean to intend they're only in plants, but they're only in plants compared to animals. They also exist in bacteria. We're not going to go into a lot of detail, but you should know that chloroplasts are the site of photosynthesis. And you should know that photosynthesis is the process of converting solar energy plus CO2 plus H2O to glucose, which by now you should know is C6H12O6. Plus, to balance the equation, it produces some oxygen. As well, plants have, and we'll stick it up here, a cell wall. And we're sticking up here because I've actually used the shape of the box to represent the shape of the cell wall around plants. They tend to be more angular or square. And a cell wall, extra layer of protection. Around cell membrane, which reminds me that the cell membrane is also an organelle shared by both. Cell membrane. And there aren't many organelles that are in animals only that we're going to concern ourselves with at this level, but one of them is something called the centrioles. And we're not going to give the details of the centrioles here because we'll actually do it down below. Okay. So I've provided you with here just two pictures that are the overview of a composite animal cell and a composite plant cell. We've chosen the word composite instead of typical because it's taking all the key parts that you might find in various animal cells. There really is no such thing as a typical cell. Okay, no typical cell. They just vary too much. Okay, so they vary in size, going from about 10 micrometers all the way up to 1 meter in length, to shape. Some are spherical, some are flattened, some are shaped like stars, some are shaped like this. We're going to talk about these ones. These are your neurons. Only turn that into a three-dimensional shape. So cells have lots of crazy shapes, so we can't come up with a typical shape either. So they vary in size, shape, components, what's found inside them, and because all of those things vary, and their function. So we're really not going to talk about a typical cell, we're going to talk about an imaginary, hypothetical, composite cell that's going to illustrate all the key features that we could find throughout animal cells. Okay. And here, just for reference, is an overview of a composite plant cell, but we're not actually going to discuss these in detail. But so you can see what a chloroplast looks like and the overall shape of the plant cell. So I said I'd like to begin working from the inside out. So we're going to begin with the nucleus. And I'm going to do a little bit of coloring here. And if you guys think I'm not stellar at coloring on the under the overhead cam, wait till you see me color on the computer. It's even more challenged. So what I'm outlining here is the nucleus in our composite animal cell. And all these features are things we're going to find in human cells. There are some exceptions. They don't exist in every type of human cell, but in most of them. So all this squiggly stuff inside here is part of the picture. And then this circle here is still all part of the nucleus. Okay. What we have over here is called an electron micrograph. An electron microscope allows us to see things smaller than the wavelength of light, which many of the features of the cell are. So this is an electron micrograph of a nucleus. And you can see some of the features here. And this is an even closer look at what's called the nuclear pores. Okay. So as we discuss the various organelles, we're going to discuss their structure, and then we're going to discuss their function, and sometimes the two sort of go so close hand in hand we'll discuss them at the same time. In terms of looking at the structure, I'm going to give you relative sizes. I don't expect you necessarily to remember the precise size unless I tell you so, but the relative size sort of order, what's the biggest thing found in the cell, what's the smallest thing found in the cell. That's the type of information you need to know. So, with the nucleus, it is typically the largest structure in the cell. And you're noticing I'm writing small. You're going to need to write small as well. There are some exceptions. First of all, in the red blood cell, because why? That's right, it doesn't have a nucleus, so it's hard for it to be the largest structure. And in plant cells. In many plant cells, their vacuole is the largest structure. Larger vacuoles. So the nucleus is defined or surrounded by a double membrane called the nuclear envelope. Or the nuclear membrane that contains nuclear pores, What do you think those nuclear pores are for? If you said they let things in or out of the cell, or out of the nucleus, that would be absolutely correct. Okay. They also have a jelly-like material, gel-like substance, that fills them, and it's called the nucleoplasm. And in the center, up here, you can see it in the picture, it's this portion here, or this really dark portion up here, and it's called the nucleolus. And it's actually stains darker or shows up darker in micrographs. The nucleolus. I'm going to ask you guys to watch your spelling because these words are related and spelling is important. And finally, you have in the nucleus, the whole thing it's supposed to be containing or protecting, DNA. And DNA can be found in two forms. Chromatin, which is the unwound form. If you think of DNA like a thread, it's like a thread that's been pulled off a spool. And this is for when it's replicating. Or it can be in its chromosome form. Which is when it's nice and neatly wound up. And that's necessary when the cell's going to divide. If it's all tangled up, all your chromosomes are tangled up in the chromosome form, it'd be really hard to equally separate them out. So it winds it up nice and neat and then divides them out. We're going to talk about overall functions. Of the nucleus. It is to protect, store, and transmit genetic material. And two, to direct gene expression. That means to determine what genes are turned on and being turned into proteins versus which ones are turned off. So gene expression determines which proteins are made, which in turn determines function of the cell. Okay. Now, with respect to specific functions of the nucleus or parts of the nucleus, we have the nuclear envelope, which protects DNA, and separates nucleoplasm from what? What's that stuff outside the nucleus, that gel stuff? And all the organelles, not just the gel stuff. What's it all called together? That's right, cytoplasm. The nuclear pores, as we suggested above, function like doors or portals that regulate what enters and exits the nucleus. So it's kind of like there's a bouncer on the door, because it doesn't just let any old person through the door. And then the nucleoplasm, that's the gel within the nucleus. Contains many dissolved components. Too many to list, but I want you to come up with a few of them. What's the big thing that's going to be sort of inside that gel? The whole purpose of the nucleus. Hopefully you came up with DNA. And then what is DNA made out of? What's the building blocks? That's right, nucleotides. We're going to need nucleotides if we're going to make more DNA, or if we're going to make RNA. And if we're going to make more DNA or RNA, what's going to help speed up the reaction? That's right. You got it. Enzymes involved in, okay, what is it called when we're making more DNA? Hopefully you came up with replication. What about if we're making RNA? That's right, transcription. What are those little guys called in transcription that recruit RNA polymerase to the site? They sort of advertise the party, say, come on over. Remember that we called them our A-listers. That's right, our transcription factors. So those are just a few of the things that are going to be in the nucleus, or dissolved in the nucleoplasm. And in the nucleolus, now the nucleolus isn't like a membrane-bound region, it's just sort of a central region in the cell, in the nucleus, where something's occurring. And the nucleolus function is to produce rRNA. And what are rRNA part of? What is it going to join up with protein to make? That's right, ribosomes, which are then exported to the, where do they work? That's right, cytosol. Very good. Okay. So that's the nucleus and all the components of the nucleus. That's probably the most involved organelle we're going to look at. It's pretty critical to the cell function. That said, most of these organelles are critical to cell function and will be found in almost every animal cell. The next portion we're going to talk about is the endoplasmic reticulum. And sort of the diagram of the cell never does the endoplasmic reticulum justice. So it's continuous and this doesn't do a very nice job of showing it. It's continuous with the nuclear envelope, and it's a set of tube-like networks that are all connected to each other, and you may not be able to see that clearly, but there's all little connectors like we see here from one tubule to the next. A tubule is just a word for a tiny tube. Okay. And this is also part of it. And the reason the diagram doesn't do it justice is because the ER is very, very thin, but it's very extensive, and it's all throughout the cell. And because we need to be able to see the other things that are in the cell in this diagram, they oversimplify it and make it look like there's not that much of it in the cell, but there really is an extensive amount of it throughout the cell. And depending on what type of cell it is, it'll have more. So if you look here, they've shown you some examples. Again, this is an electron micrograph. Of the endoplasmic reticulum, ER. And you can kind of see examples of it, these little tubules coming through here. But it's really quite messy if you look at it closely in a cell. It's highly organized, but there's just so much of it folding back and forth throughout the cell that it looks kind of messy. In fact, that's what its name, endoplasmic reticulum, represents. Endoplasmic for endo meaning in, plasmic, the plasma, in this case the cytoplasm, in plasma, and to reticulate means to bend or fold back and forth. So it just is referring to this membrane structure that folds back and forth throughout the cell. So structure of ER is it's continuous, meaning it's attached to, with the nuclear envelope. Size-wise, it's really difficult to describe how big it is because it's very, very thin, fine, sort of like the way a spider silk is fine, but very, very long and twisted throughout the cell. So it doesn't have a particular size. It's continuous with the nuclear envelope and is a series of interconnected tubules. That stretch throughout the cytosol, or throughout the cell. And there's actually two types, and they're described based on their structure, but I'm going to put it down under the function because each type has a different function. So the first one is the rough ER, or RER. And it's called rough because if you actually look at it under the microscope, it looks rough because it is studded with ribosomes. And the ribosome, what is the function of them? That's right, they make proteins. So ribosome forces protein into the ER space. So these are tubes, so it's into the center of the tube. As it is made. And these proteins are actually going into the rough ER because they have a particular destination to go to. So these proteins are destined for one of three outcomes or locations. Either a particular organelle, the plasma membrane, which is the cell surface, or secretion outside of the cell. So they're actually going to be delivered to the outside of the cell. And we're just going to continue the rough ER down a little bit lower here. So the rough ER allows for directed transportation of proteins. So it basically controls where the proteins are heading in the cell. And some protein modification. So as the protein moves through the ER, changes are made to it. It's improved. And some examples of this are glycosylation. Glycosylation is a fancy word of saying we're adding carbohydrate chains to the protein. And those carbohydrate chains can perform a variety of functions. And we'll talk about them specifically in different areas of the body as we get there. And you will find high levels of rough ER in cells that are secreting proteins. So not all cells have equal levels of ER. An example of cells that are secreting proteins are such as B cells that are making antibodies and secreting them into our blood. Okay. Now we have to talk about the function of the smooth ER. The smooth ER is called the smooth ER because that's what it looks like under the microscope, because it lacks ribosomes. It has a very different function. So the smooth ER functions include three things. One, it produces lipids, including phospholipids for membranes, because as your cells grow, they need more membrane. And steroids, such as testosterone. And they also detoxify drugs. So for these guys, you find smoothie R in high levels in cells secreting hormones, because remember, hormones such as testosterone really are just steroids, which are really just lipids. Such as special cells called interstitial cells. Of the testes, where testosterone is being produced. And I don't expect you necessarily to remember interstitial cells now, but when we get into the reproductive system, we'll come back and talk about them. Okay? So that's the smooth ER and the rough ER. Now at this point, I think it'd be a good idea to sort of compare what the different functions or different structures of the cell do to an analogous system, because there's lots of complexity in the cell, and if we can compare it to some other system we're more familiar with, it's a lot easier to remember. So we're going to introduce some analogies here. So, and our analogy I'm going to choose for this lesson is that the cell is like a car factory. The cell is like a car factory. So if the cell is like a car factory, then the ER is like a conveyor belt. In the assembly line. Where components of the car are assembled and modified as they are moved throughout the factory. And if we're going to do that for this organelle, we probably should do it for all the organelles. So maybe we should go back up to the nucleus and do that. Okay. So the nucleus, if the cell is like a car factory, the nucleus is like the control center and factory manager. So the control center has the computer that stores all the information, and the factory manager decides how that information is used or applied. So stores and applies all info necessary for running the factory. Okay. So we're just going to pause there. You can take a break. Where you should get to or where you should take your breaks will actually be dictated and communicated in class. We may do most of this in class, but there may be some that you have to do at home. If you have any questions, because you are watching this at home, please come see me in class. Otherwise, have a good day.